I guess I'm going to bring a quite different view on systems, which is more molecular, more biochemical. And it has to do with the structure of the molecules. It has to do with the structure of proteins. So I will give just a brief introduction that probably you all know very well from basic biochemistry, which is, what about the structure of proteins? Proteins are polymeric molecules formed by amino acids. They call primary structure just the chain. Then it folds in the space and generates regular secondary structures. Alpha helices and beta strands are the typical ones. These fold on a tertiary structure and make the interactions forming quaternary structures that bring us on function. Now, knowing the structure of proteins takes a big effort on crystallography. So that's the reason that along the years we have increased on the amount of sequences that we know of proteins, but not much on the structures that we know, this orange line. But there is a good news. The good news is that if we look along the years, how many new folds on different structures have appeared. It happened at the very beginning we had many, but then after the years, we are dropping down to a few. And this is a very good news, because that means we can make predictions. We can predict things with the things that we have already seen. Now, we have heard about modeling and models, but what's a model? Now, this is a plane. Well, actually, this is a picture of a plane, a real plane that flies. It has some bombs to throw, some pilot and a co-pilot. This is a model. This model looks exactly the same as this one, or very much the same, with the cockpit, the pilot, the wings, everything you want, but it doesn't fly. Now, that sounds a model. It doesn't look like this, but this flies. Both of them are models. I want you to recognize that, you see, both of them are useful. It depends just on our focus. So depending on what we want to see, we will use one model or the other. So actually, for protein structures, we are going to do something called homology modeling, which is based on the similarity between the sequences. And then we take one template for which we know the structure, and then we just align the sequence in red we want to know, and then repeat exactly the same distances that we have already seen to reproduce a model. Finally, we have to validate our model using some kind of energy, and some of these energies are based on the Boltzmann energy. So it's called a Boltzmann Law that relates or associates the probability of a microstate with the energy. In our case, will be the probability of two residues being in contact will bring some energy. And this probability can be taken from what we know in structures. Now, on the talk, I will go through three different parts. The first part will be on the role of the tertiary structure. Then I will on systems biology or systems medicine. Then I will go on part two with the role of quaternary structures, that's on the interactions. And then in the last part, on the development of several bioinformatics tools that help us to understand the systems. So for the part one, on the role of tertiary structures, first, let me go a little bit more deep on our knowledge on protein structure. There are secondary structures like betas and alphas. These are very regular, but there are also the connections between these secondary structures, some kind of loops we called, between betas and alphas, alphas and alphas, betas and betas. They have also sometimes some kind of regular structure. And they also form some kind of shape in the three-dimensional space in the way we place betas and alphas. So we can classify them. It's not as easy as an alpha helix or a beta strand. We classify it depending on the kind of loop, so it's an alpha-alpha or an alpha-beta or a beta-alpha. Then the length, it's just based on one residue or two or three or four or 40. Then on the shape, how is the topology? How do they look in the space, the alpha and the beta or the beta and the beta? And also in the Ramachandran conformation or 5-SA conformation of the amino acids in the loop. Now, also long years, there is some kind of plateau that it's also reaching a way in which we are repeating the same kind of conformations. And it has given us some kind of idea, a few ideas about what amino acids we're using. For example, one of the keys is that Residues charged with positive charge, like arginine or lysine, usually they are found on the C-terminal tail of an alpha helix. And residues with a negative charge, like aspartate or glutamate, they are found on the N-terminal tail of an alpha helix, and they stabilize, they elongate the helix. And this is something we have seen with many different structures. Also, we can redesign the structures. So just by knowing what's the location of an alpha or a beta, we can find in the database several other structures and then place them on these secondary structures just to reproduce a different way or a different function on the protein. So it's a kind of redesigning or engineering of proteins. So let me go on this first example with proteins, with the structure of proteins and their role on system biology. I will take the example of PARP1. That's an enzyme, a particular enzyme. Here is the system. The system is the following. It's a cell, and the cell has in the nucleus, the DNA is compact by nucleosomes. These nucleosomes have mostly positive charge, and the DNA is just surrounding these histones, this nucleosome, and then just by this, it gets compact, and the genes that are could be expressed on the DNA are not expressed because they are, compacted by the nucleosomes. You need to uncompact this nucleosome in order to express a gene. Also inside the nucleus, we find some other elements like this one, NMNAT1. NMNAT1 is an enzyme, and this enzyme takes ATP and NMN. Now this is ATP and this is NMN, and it produces NAT. You see it's very similar structures here. The only thing is there is one pyrophosphate exceeding that it goes out, the pyrophosphate, and then you get NAT. Now, PARP1, this enzyme I was telling you, uses NAT, this is structure, and then it splits in two parts. You see this one can also be split in these two parts, ADPR, adenosine phosphoribose, and the nicotine. So adenosine phosphoribose can produce a polymer called PAR, polyadenosine ribose. So it's a set of polyadenine riboses, similarly as the DNA, has highly negative charge. What happens is this. This accumulation of PAR will also interact with the region of the histones and then release and then activate some genes. So it's very important, the attitude or the activity of PARP1 on the activity of many genes. Now, the activation that is produced in some genes, in the case of progesterone, is the following. It should act in a way that activates some genes. Now, the question is, it may increase the quantity of PARP1, and that increases the quantity of PAR, and that releases some gene. What happens is that when looking at these images, we can see that PAR has increased after 15 minutes, but PARP1 has not, which means that something else is happening here. We can see that NAT, the substrate of this enzyme, is dropping down. So it means that the enzyme is more active, even though it has no more quantity of the enzyme. So let's go a little bit farther on the system. What happened with the progesterone? Progesterone activates a CDK2, a kinase. This kinase phosphorylates PARP1. Now, we know where it's going to be phosphorylated. It's in these two serines, just by looking at different side-directed mutagenesis, changing the serines by alanines, we can see the change and then how it's differently acting. So we have a phosphorylation of PARP. This will produce some PAR, polyadenine ribose. Then this will activate by binding the histone, adding some negative charge, releasing the genes. What we don't know is what has happened here. And this is where our knowledge on structure helps. Because this phosphorylation happens in the N-terminal or close to the N-terminal of an alpha helix. And I said before, that's something we have seen in molecules. By adding negative charge in the N-terminal site of an alpha helix, we extend the helix. So the helix is longer, and what happens if we extend and we modify the helix and we elongate it? And I want to show you here what happened. In green is the actual model of PARP, and here inside, in this cleft, is the active site, where it's going to be bound NAT. When we elongate the helix, we modify the location of this domain. And now the active site is more open. So it's easier to get NAT into the active site, and then it speeds up the reaction. So it's producing more ATP and more PARP, sorry. Let's see some other examples on quaternary structures, because these usually are the more typical cases relevant for systems biology. How do they act in protein-protein interactions on the system? There are different web services that will give some information to us about structures of protein-protein interactions, Interacton 3D and 3DAD or PRISM as the typical ones. So I will base on these few examples. One is the ATP synthesis in the nucleus. Another one example is the retinoblastome activation. Then I will go into something more close to system medicine on protein networks, and then act with or try to infer comorbidities. So let's go to the first example, just going back to the previous one. We have seen the work of PARP. Now, this is producing this polyadenine ribose, and this is kind of collecting a repository of ATPs in the form of PAR. The thing is, ATP usually is the way you get energy on the system. You get the ATP from mitochondria, but if you have to get the ATP in this way and then get it into the nucleus, this is very slow. If something has to happen very quick, very fast, you need energy quickly, and you have to obtain the energy very fast. Now, can we use PAR to obtain this energy? Now, trying to answer this question, we were looking at some enzyme that could just perform the action of transforming PAR into ATP by first moving into ADPR and DADPR to ATP. So in the group of Miguel Beato, who were doing all these experiments, came to our lab again, trying to find out if there was some explanation to this, and they came with this molecule, Nodix5. Now, Nodix5 is a dimer. In this cleft is where it binds the ADPR, okay? Now, this cleft is very, very tiny. It's difficult to place the ADPR plus pyrophosphate to make a reaction and produce ATP. So if we opened this dimer and release this part, you will see how it is located, the ADPR, and the difficulty on finding pyrophosphate getting into this cleft. It's easy to make water to pass through, but not a pyrophosphate, which is a bigger molecule. So if we want to activate this, what happens is that it has to change either the shape or the dimerization. And what we know is the following. In case of a dephosphorylation, it's producing ATP. In case of a phosphorylation, it's producing AMP. So in this case, or in a phosphorylation, water gets into the cleft. In the case of a dephosphorylation, pyrophosphate gets into the cleft. The only explanation to do this would be to break the dimer or change the structure. And if we have a look at the dimer, this is the residue that gets phosphorylated. It's a threonine. It's very close to a lysine in the other side. Now, a negative charge of a phosphate here interacting with a positive charge in the lysine here stabilizes the dimer. If you dephosphorylate this, that's a destabilization of the dimer, and that opens into a monomer, and that changes the activity, getting ATP instead of AMP. So that would explain the process. So actually, in the lab of Miguel Beato, they did several tests. They tested the phosphorylation of T45 and the dephosphorylation with the action of progesterone. They prove the fact of the dephosphorylation. They also test the activity, and they test by mutagenesis, side-directed mutagenesis, what happens if you change the threonine by alanine. So it happens that the dimer is not formed, it's acting as ATP all the time. What happens if you change the alanine by aspartate? So it behaves like a phosphorylated threonine, so it doesn't produce ATP, it's only producing AMP. So that would explain, and also, just theoretically, we could calculate the interaction of the two monomers, and they could test experimentally with thermophoresis and microscale thermophoresis the formation of monomer thanks to the dephosphorylation. Let me go to a second example in which the knowledge of three-dimensional structure will help. That's the activation of some genes that goes in the cell proliferation of yeast, and it's produced by this case. This retinoblastome is binding transcription factor DP1-ETUF. Now, when it binds in here, this cannot bring polymerase, so the gene is off. If we release the retinoblastome from this binding, then polymerase can get in, and then the gene is on. What happens is this. It comes a phosphorylation by CDK, and this phosphorylation of retinoblastome will produce the release, and then it produces the cell proliferation. The weird thing here is that under osmotic stress, another kinase, P38, so it's just an over-phosphorylation, produces the opposite effect. And the question is how? How is this produced? So let me go to what we know from the structure. This is the structure of ETUF-DP1, and this peptide here is what we know from the structure of retinoblastome. It's part of the C-terminal site of retinoblastome. And if we look at the surface of DP1-ETUF, it has one region which is highly negative and one region which has more positive charges. I want you to locate now the region where is the serine that comes phosphorylated. It's just here. And here it's a glutamate that it's acting with the positive region here. So this serine, when it comes phosphorylated, increases the negative charge. Now what we have is a repulsion between two negative charges, and then it gets released. So dephosphorylation will act on the release of retinoblastome from DP1-ETUF, and then the activation. We can see this just by looking at the energies of the system with the model, and then comparing what happens with the phosphorylation. But what we don't understand yet is, okay, what about an over-phosphorylation? How this produced, again, a binding. So the next thing is maybe a different molecule. So what we did was, okay, we took this peptide and looked in the database for other proteins that contain something similar to this peptide. For our surprise, what we found was exactly another region, now in the N-terminal site of retinoblastome, which was this one. And it was very similar. So what we did was, okay, so we modeled this N-terminal region based on the C-terminal part, and then tried to see what happened just theoretically with the energies. So what we tested was what happened with this region with the same kind of a structure, plus the phosphorylation of serine 252, which is this one here, and another phosphorylation for serine 249. And what happened is that the energy drops down, so it's binding again. So the over-phosphorylation produces again the binding of retinoblastome, but with a different region, the N-terminal site. Obviously, this needs to be proved experimentally, so this was in the lab of Laia Nadal and Frances Posas, who did it. So they took the first 300 residues of retinoblastome, and they tested what happened with different mutations. Either they put alanines instead of serines, or they left the serines, and they found that certainly these were the residues that get phosphorylated thanks to P38. The second thing was about binding, and what they saw is that in the case of osmotic stress, when they have an aspartate instead of the serine or the threonine, so it becomes active all the time, so it's acting with respect to osmotic stress or without osmotic stress, always the same. So the behavior is the same, plus they prove the binding with co-immunoprecipitation of ETUF in the case of in the excess of P38, so producing the phosphorylation, recognizing the binding of this N-terminal site with ETUF and DP1. So proving that the example was applicable, that this was a real model for us. Now let me go to a different model. We have seen very teeny details, so how it works with the small molecules. Let's go for a larger system. Instead of talking about just single protein-protein interactions, we will move to networks of protein-protein interactions. If we take the interactome, the whole set of protein-protein interactions, and then we map different genes and mutations that have been associated with the cis, we have some kind of new map for this network telling us the information of who or what is connected with who. Now, with this, we use an approach called guilt by association, that says that if you are connected with neighbors that are associated with cancer, probably you are also associated with cancer. Now, this concept works through the network in a way of transferring the information on a first step with your neighbors, but then you yourself, a node has changed its own behavior, so it's acting on the other. So it transfers information. It's a message passing. There are many different programs that use message passing. One of them is in Google, by the way. It's the one that we used for transferring information through the net. So we can apply some of these methods that transfer the information and then recognize many other genes associated with a disease, as long as we have the network and then some seeds for assigning a node on a disease. So we apply this on the case of brain and lung metastasis. So we use some information on genes associated with either brain metastasis or lung metastasis, and then generate the two networks and then transfer the information. And what we saw was a connection of several genes with some others that are totally new. They have not been associated with any of these, with metastasis, brain or lung metastasis. They were new for us, and they were connecting some of these other clusters associated with one or other disease. Then we can go to automatically find the structures of some of them. And by doing this, we have some extra information about what are the interfaces and the role they are playing. And the very interesting thing is that we can go a little bit further and find some proteins that are associated with two different proteins, one associated with brain, another one associated with lung, and then some mutations that are affecting on these interfaces, which tries to explain in a molecular way how both metastases can be associated or related. And this brings me to a view which has to do with comorbidities. Can we analyze comorbidities of diseases by applying our knowledge of the network? So here is a plan. We are going to take information from protein-protein interaction networks. It's different data sets on protein-protein interactions that we have in different releases. We use Biana. It's a program, it's a platform that introduces information from different experiments. Then we used another program, it's a platform, DisGenet. DisGenet associates diseases with genes, so we can infer a mapping. We can get a mapping on the network. And then I'm going to use two different versions of DisGenet, one from 2014 and another one from 2018, with the idea of validating the results. So we will see how. And the idea is the following. We will use guilt, this transfer of information through the net. So with some initial seeds or genes associated with some disease, we will see what happened on the network. And if at some point we have an overlap between two diseases, thanks to this expansion. So the idea is the following. First, we have some exploratory set, some genes associated with the diseases, and then the expansion. And then a validation set, some genes that we know 4 years later has been associated with the same disease. So here it's a validation set. There are just a few in which we can apply this, not for all diseases, but for many of them, at least 252 diseases, we could apply it, this approach. And the approach does the follows. We have this overlap between the genes or the proteins associated with the two diseases is this intersection here. And then we go to the validation set and check what is our intersection if we are really finding the same ones. Now, this can be calculated with some probability. So with respect to random, using an hypergeometric distribution and then some testing, multiple test p-value correction by Bonferroni or FTR, and it will tell us how good we are on associating the two diseases. The same as can be done with the overlap of genes, can also be done with the association of functions. So we can use the functions of the genes that comes from the gene ontology using molecular function, or gene ontology using biological processes or pathways, reactome. So we can have an overlap of functions. I will not get into the details of two different ways to do it, but it's just, instead of overlap of genes, are they common pathways associated in two diseases? And after that, what we have is a set of several connections of diseases with relevant or significant probability. So if we look at them, we can make clusters. And once we make clusters, we have something like this. It's a new network. We started from a network of protein-protein interactions. We end up with a network of diseases. Diseases that have become associated because of the protein-protein interactions. Here is a set: asthma, pulmonary diseases, arteriosclerosis, hepatitis, arthritis rheumatoid. Many of them are associated with functions common of inflammatory processes. So it makes some sense. Actually, something that has been already known, this association between asthma and arthritis rheumatoid is one of the more comorbidities that comes into different works. A very interesting one was this cluster here, because we have leukemia, lung neoplasm, neoplasms, colorectal neoplasms, carcinoma, Alzheimer. And that's surprising because, in principle, one thing that, this has nothing to do. But it happens that already some publications in the 19s were saying that there was an inverse comorbidity. So, just taking away the cases for the age, because there must be there can be a correlation with age. So you just take away this possible link. There is an inverse comorbidity, means that those having cancer don't have the tendency to have Alzheimer, and those having Alzheimer do not have the tendency to have cancer. So that will be a very interesting case for trying to analyze more deeply what's going on. And trying to understand more deeply what's going on means, as everything has come from our knowledge on the molecules, on the interactions between proteins, can we now map these interactions and try to understand the system? So what we did was the following. We have these connections between diseases. We have the genes that make the overlap, so the proteins that are connected. And we can see many of the protein-protein interactions that are associated with these common diseases, and then try to analyze the structure of them and do it in an automated way. To do that, what we did was first make an alignment of the sequences of the proteins with the databases for which we know the structures coming from PFAM, and then recognizing the interface, and from the interface trying to infer if there were some mutations associated with the diseases found on these interfaces. This comes from ClinVar. And thanks to that, for some of these comorbidities, we can find some interactions that are highly associated with some mutations in the interfaces, highly associated with this comorbidity. This is, in the case of rheumatoid arthritis and liver cirrhosis, is a metalloproteinase inhibitor 2 interacting with ubiquitinous collagenase type 2. The one I mentioned before, rheumatoid arthritis and asthma, associated with tumor necrosis factor. This has been actually used as one of the targets for many pharmacs, for many drugs. But let me go to this particular case, Alzheimer and cancer. What kind of relation we have here? If we look at the association of the functions, we mostly found these apoptotic processes, so apoptosis. And if we look at the genes, we find these two, BAX and BIT. Now, BAX and BIT are very relevant to produce the apoptosome. Is one complex that produced the apoptosis. BAX has been associated with Alzheimer's disease and also with some cancers. And we also have some mutations for this. This mutation, glycine 108 to valine, has been associated with Burkitt lymphoma. And we also have the structure. And if we look at the position of this mutation, it happens that it's really in the interface between Bax and BIT. So the thing is, what about this inverse comorbidity? Well, if we are affecting this apoptosome complex by inhibiting or by activating it, it will have a different behavior. So if we inhibit the formation of the apoptosome, what we are avoiding is the apoptosis. And this helps in the case of cancer, because cells will survive. On the other hand, if we do the other way around, if we try to reinforce this apoptosome, what is going to happen is that we will have apoptosis, and then what we have is the death of the cell. That's what happened in the case of Alzheimer's, where the neuronal cells are dying. So it's an inverse comorbidity in this sense, and both of them are associated with this process, apoptosis and this interaction. So we can further go into a modeling of this interaction, and then not only this mutation, but also many other mutations on this interface can also be relevant on the interaction and in the formation of the apoptosome. This one has also been associated, the arginine to leucine, with cancer. The other ones are not yet known, so have not yet been associated. They are in ClinVar, but the association is not yet, it's not strong enough to be associated with any of the diseases. But that's the information we are extracting. We go deep into the molecular system, try to figure out what's going on. So there are many different bioinformatic tools that can help us to produce all this kind of analysis. The first one, it's trying to figure out from the network what are the protein-protein interactions relevant on some of these diseases or in the comorbidities. The second one is try to automate the modeling of the protein-protein interactions, and then try to infer some kind of association when we don't have a structure. So let me go first to the program Gildify. This is a program we developed just with the idea of analyzing networks and associating new genes with some diseases, and then also try to analyze comorbidities. So in that case, you can place your sequence or even just the name of the gene. The species can be Homo sapiens, or you can be testing a different species like mouse, the tissue. And then with the network associated with the tissue and the species, we can get a new network associated of genes. And then with two of these associations, we can have an overlap of them and check the comorbidities. And the overlap can be done either by genes overlap or by functional overlap. Actually, we also have some pre-calculated sets, and these pre-calculated sets have been obtained with some diseases, using the seeds of these diseases, targets associated with them, but also with drugs. So what we have with this is a set of candidates that are also associated with some drug, and then we can just merge both. So here is an example in which, for example, these drugs have been used for non-small cell lung carcinoma. You can see that the p-values here of association are very high. And the interesting thing is that some of them have also a very good p-value for applicability on breast cancer. Actually, this one, palbociclib, is recently being used in some clinical trials for breast cancer. So it's known for non-small cell cancer, lung carcinoma, and is now being used for breast cancer. To automate protein-protein interactions, we have also developed a program, ModPin. We don't have a server yet. We only have a GitHub to download and then execute. The idea is to get the set of sequences, search on the database of protein structures, get the pairs, model them, and then analyze by Rosetta with the energies, and then select the common, select into clusters, group into clusters, those having common interfaces, and then analyzing the energies of the system. So here is an example using data from mutations in SCEMP that has been associating the delta-delta G, the difference of the free energy, the affinity, with the scores on energies that we can get using different programs, like this one is Rosetta, another one is ZetaRank, another one is Voltex, classical programs to calculate the energies. But what we can see here is that the correlation is not very good. What happened is this. We have different conformations in the data set showing us how two proteins can interact. So you have more than one interaction. Now, that means that we are mixing everything in this, and then this formula that comes from free energy perturbations, that comes from physics, from Zwanzig's. This only works when we have a very close system. If the conformations of the space are too different, this doesn't work. And this is what happened here. It's not working. So, in a similar way as I did before with the case of Bax and Bit, we can analyze when we have exactly a very similar interface, what's going on. So, and here are different mutations, and you can see how significant are the distributions of the energies. And these mutations occur on the interface. So we can just select. If we select very careful the sets of these conformations of protein-protein interactions, we can see now when the mutation happened in the interface and if the mutation is going to be significant or not for the interaction. So by grouping this, we can just select some of the protein-protein interactions and then check again. Now the correlation looks nicer. The only thing is that you have to be careful on this selection. Now, every correlation that we are obtaining here is associated with some parameters, like what's the significance of the difference between the mutant form and the wild-type form, what's the percentage of the mutation in the interface, was this the most populated cluster or less populated cluster. So by doing this, we can just select these parameters according to how good is the correlation and then apply our set of predictions. Now, how good are we predicting the change of binding? So is going to be broken the interaction or not? Now, with different Pearson correlations, we have different parameters. For a few cases, we have some parameters that obtain a highly success. So that means that we are able to predict, although not with a very good coverage, some cases of the effects of mutations and then decide whether the mutation is going to break or not an interaction. Obviously, this has been applied within the same set. So one of the things that we can complain on this is that you are validating on top of something that you have in training. So what about a different set? So we use a different set, the set of protein-protein interactions that have been disrupted, and it's in EBI. So these different sets, mutation decreasing binding or mutation disrupting interaction, and we test with the same Pearson correlation or parameters that we tested before in SCEMP, and then we still keep a high success ratio when we have some of these values of Pearson correlation, so the coverage is not that bad. So in principle, we can apply this as a way of predicting the effect of mutations on protein-protein interactions. Now, to do this, we need the model, the structure of the protein-protein interaction. What happens if we have the structure of the two proteins, but we don't have the structure of the interaction? So in this case, the way to act is usually by docking. So what I'm going to show here is what happens now if I try to find the interaction in the near native phase, just close to the real solution, or in a phase, so it's similar surface but not exactly the right one, or back, so the surfaces of the interaction are looking away each other, or phase back, which is half and half. And if we look at the energies of this is obtained with a data set for which we know the structures and we know the energies, of course. We have this plot. So once we model the near native, we are getting a good result. But then we are facing with a phase or phase back or back, the energies are up here, so they are not good interactors. This makes sense. But the very interesting thing is that if I look at the correlation of the energies. So what's the average energy of a phase-phase with respect to a near-native, or with respect to a phase-back, or with respect to a back-back? So here is a near-native compared with a phase-phase. You see some correlation. And here there is also some correlation between the phase-phase and the phase-back, or the phase-phase and the back-back. So that's interesting, because what it's telling me is that if two proteins have to interact, they will have a very good binding, not only in the region of the binding, but also on the other sides. On back-back, so totally different region, better than others that do not interact. Now, why is this happening? Well, one of the reasons is that if two molecules have to find each other in the space, they need to collide. And for colliding, it's very difficult. They have to find each other. Now, once they find, if they bounce and then try to find again, it takes some time. So this is very slow. So the best way is, once they find, if they really have to interact, the best way is that get close, don't get away. And then at some point, just move around until they find the real solution. And this is what happens here. So what we see is that those molecules that have to interact, they really have a good binding, better than others that don't. So those with a strong affinity will bind in any of the phases. Doesn't matter if it is the right one or not. And we can use this because if you take just the average of the binding, the independent of whether you have found the right conformation for the binding, you can use the average of the energy just to predict the delta G. And this is our predictor and the correlation, which is rather good. It has an error of around 3 kilocalmol, but it's a good predictor. Finally, what happens when you don't have the structures? Can we do anything? So this is another program we have developed. It's called IFRAC. And what it does is the following. If two proteins interact, I want to know where is the region of the interface? Where are they interacting? So what we do is the following. We take one of the queries, one of the sequences, and then search in the database of BLAST, all the protein sequences, and then align them. And then I do the same for the other protein, query 2. And then I select those that I know they interact. And by selecting those that I know they interact, I extract some kind of pattern, and then I see that some regions are common in all the different sets. For example, this region here has appeared in this, and this, and this, and this, and this region has appeared in these other ones. So maybe that region is the relevant one for the binding. So by doing this, I get some kind of a score that is telling me what is the region of binding between the two proteins. These white dots here are from a real structure, so it's the real binding where it occurs. And this red one here is the one that gets the higher scores. And we have used this in some particular case, is the case of the A-beta-40 peptide. This is an alpha-beta amyloid. And this peptide interacts with albumin and with clusterin, and we can check what are the regions. And then from these regions here, we recognize some pattern for the alpha-beta-40, and then some regions for the clusterin and for the albumin. And if you look at these regions, they look very similar in sequence too. And this is telling us that there is a particular region that binds with the alpha-beta peptide. And what we did was the following, was to produce this peptide and then test what happens. And this is the albumin C-tail peptide. When you put it in the presence of the alpha-beta amyloid, then it's not produced the fiber. So it produces the interaction and breaks the fiber, which is quite good in the sense that it's one of the problems in the case of Alzheimer. We also model the structure by taking the structure of the alpha-beta peptide and the structure of the albumin and then modeling one on top of the other. So we also could advance in some of the regions and get some of the bindings, helping us to detect the way it interacts. That may help also to produce a new drug for breaking the formation of fibers. Now, all this work has been, as you can see, produced with many different labs that do the experimental job. So I have to thank Miguel Beat and Ronnie Wright for all the work on PARP and NUDIX, and also the groups of Francesc Posas and Lion Nadal for the case of retinoblastoma, and the group of Fran Sanz for the use of this genet. So here are the grants that we have been using, and this is just a platform where we have most of these programs, and if you want to just click on them, and then you can go from the protein interactome to protein-protein interactions, particular sets, or mutations, and so on. So thank you.
So thank you very much, Baldo, for this great talk on the very lower level of modeling, which is actually then the basis of everything that has been further discussed in this summer school. Any question? No? Do you have hungry mutation? Yeah, Marco.
There is a large explosion of the protein structures with the cryo-EM these days. So does it help you a lot?
Yep. For cryo-EM, for a very long time, had a resolution that was around 6 angstroms. That was not enough. But it has reduced now up to 4 angstroms. Now, this helps in quaternary structures a lot, because even if you don't get all the details, you can get the details from crystallography and then plot, or just superpose the structures that we know on these other macrostructures, these maps from cryo-EM. And this is really helping a lot, yeah. And in many senses, in the senses of mutations that you can find on the interfaces, in the detections of the structures that we don't know anything yet. So, yep. Thanks.
Yeah, I have a question. That's curiosity. So when you pass from the protein structure model then to the interactome, so in order to go then to more general level of disease mechanisms and associations, so we've played with interactomes for specific diseases. So in Roma, and what we've seen is that when we use bioinformatic tools then to generate the interactomes, we have something that is too unspecific. And we are absolutely not able then to capture what experiments are seeing. So proteomics, so high throughput proteomics analysis, or even gene expression measurements. So, but when we use very reduced corpora based on literature search, I mean maybe 80 papers, journal papers, so we're actually able to get more precise idea of what happens. So here is a question. So what then would be the impact within your full pipeline, so of the unspecificity of the corpora?
Yeah, this really depends on the focus you are using. The thing is, if you go into the details and you're going to model the system with, let's say, with some standard equations of differential equations and then try to see the model, you need very few proteins and genes and just very detailed. But if you want to go through, try to figure out when you don't know anything. So you're looking at the papers, but what about if you don't have papers? You don't have this information and you have to go through everything. Then it's better to use the maximum information you can get. The problem with the maximum information you can get, which is the case of the protein interaction networks, is that you are using all proteins that you know and all the interactions that you know. But what happens if some of these proteins have not been expressed? Or they have a different isoform, or depending on the tissue or the cell, is a totally different set of interactions. You don't know this. So you are transferring the information just with the whole set that you have. And you don't go into the details. So that's the kind of thing that you are missing. And if you go into the details, who is expressed at the right time, then obviously the network drops down to a very few, and then these are the ones that you can analyze.
My impression related to that is that there are very highly multifactorial diseases that very often are even not classified as diseases, but just disorders.
Yeah.
And that's a fight that many people have, no? And so in those diseases, very often, so the real activity of the cell at the gene level or proteomics level, so depend on external factors. External factors such as mechanical loads, osmolarity of the medium, etc. Which means that though some protein interactions so might happen for this cell, for example, in the bench, so in the lab, so in the real tissue, then you might have some inhibition due to.
Yes.
So is it possible somehow to try to use another kind of knowledge in order to filter this information, at least conceptually speaking?
This is what we have tried when you're seeing here. When you can select the tissue that you want to use. But, for example, when we are selecting here the tissue, we are taking information on RNA, what's the expression, and we are taking information from the mapping that we have, the interactome mapping, that comes from immunology. It's called immunoprecipitations. So it's not very clear. It's not very neat, and most of the information is not very neat because tissues have different cells. So you would really need particular cells of each one of the tissues just to do a proper modeling. So it's very difficult in that sense, and there is no information to do this. That's a problem. Even when you get information from microarray expression, it's a mixed. It's a mixed sort of the tissue that mixes everything. And nowadays, with single cell experiments and with analysis of long RNAs, then probably this is just improving. But yeah.
Any other question? No? So then I propose so we close the morning. And so let's go for lunch. I don't know, what time is it? 2 o'clock? Yeah, 2 o'clock. So maybe we'll delay a little bit in the start of the hands-on. So to, yeah, at the latest 3 o'clock. If you can be at 2:45, that would be better. Okay. No, hands-on are in another building. I will guide you through. Yeah.